Hello, everyone. My name is Aaron Rodrigues, and I'm a PhD candidate here in the Department of Entomology at Purdue University. And today I'd like to speak with you about my dissertation, molecular and proteomic characterization of Blot-2 allergen in the German cockroach, Blotella germanica. So I'm going to start off with an outline. I'll go into my introduction and also different chapters. So I'll start with my hypotheses and objectives, my chapter 2, manuscript 1, objective 1, my chapter 3, manuscript 2, objective 2, and overall conclusions. And I'll finish up with future research goals, as well as my acknowledgements, and wrap up with a Q&A session. So for the introduction, urban entomology is the study of insects that frequent human habitats. Examples of these are termites, bedbugs, and cockroaches. I'll be speaking about cockroaches today, and ideally cockroaches have over 4,600 species, but less than 1% of these are pest insects. So less than 1% of these actually pose a threat or are pests to humans. And the cockroach that I'll be focusing on is the German cockroach. And here's a brief description of the life cycle. So it starts out as an egg. The eggs are produced in a case called an ootheca. They then hatch and become nymphs and develop into adults. So they can develop into either an adult female or an adult male. Here you can see that there's a little bit of sexual dimorphism going on. So the male has a relatively narrow abdomen. He's a little bit smaller than the female. The female is a little bit girthier and has a wider abdomen. The female will actually carry this ootheca around, and when the eggs are close to hatching, she'll deposit it typically on the ground, and they'll hatch from the ground. And the scientific name for the German cockroach is Blotella germanica. They belong to the Blotella genus, along with a few other members. So the German cockroach does economic damage. So it damages different food items. It will consume food items, infect the food items with bacteria, deposit feces. They're also aesthetic pests, so they'll deposit feces on different household surfaces, and these tend to be aesthetically unpleasing. German cockroaches are also medical pests, and so they produce a number of proteins that are allergens. They act as allergens in the human immune system. So Blot-4 is an example of a German cockroach allergen, Blot-5. And the focus of my dissertation was actually a protein produced by the German cockroach known as Blot-2. So with that, I'd like to go into the hypotheses and objectives of my different manuscripts. So for chapter 2, my hypothesis was that Blot-2 mRNA and protein are expressed in multiple tissues and life stages with relatively high expression in the male Tergal glands. My objectives for this were to determine the relative abundance of Blot-2 mRNA and quantify the expression of Blot-2 protein in different tissues, sexes, and life stages of the German cockroach. So my chapter 3 hypothesis is that there are multiple protein and allergen isoforms active in the Tergal glands, and my objectives are to determine the identity of proteins expressed in the Tergal glands and also determine the identities of proteins that co-immunoprecipitate or interact with Blot-2. And so I'll go into these in depth later. For my manuscript 1 or objective 1, I submitted the manuscript to the Archives of Insect Biochemistry and Physiology. And also I've done a series in which I sort of categorize the protein or gene expression of Blot-2. So the title of the manuscript is Expression Profiles of an Inactive Aspartic Protease, Blot-2 allergen, in different tissues and developmental stages of the German cockroach, Blotella germanica. And to begin sort of describing this paper, I will have an introduction into the Tergal gland. So these are structures that are found in the German cockroach, but they're also found in other species of insects. So the Tergal glands can be found in honeybees, and they lead to ovarian activation. They can be found in bumblebees. So the Tergal glands in bumblebees secrete pheromones, and these pheromones stimulate the bees to travel farther from the nest to forage for food. Also, Tergal glands in the German cockroach are involved in courtship. So in German cockroaches, the Tergal glands are glands that produce a sugary substance, and the female will consume from the Tergal glands, and while she does so, the male will attempt to mate from her. So one thing that's important to note is that the Tergal glands in German cockroaches are structures that are only found in adult males. So you'll notice that in the experiments I highlight later, Tergal glands will be included in every replicate that I do. That's simply to act as a positive control. They're only found in the adult males. And so there are structures that produce sugars, proteins, fats, lipids, and other substances. And the female is drawn in by these and will consume from the Tergal glands. And while she does so, the male will attempt to mate with her. And also, Tergal glands are used outside of the context of reproduction. So the females will eat from the Tergal glands even when they're not mating with the male. And also other members of a German cockroach group or intrusion will eat from the Tergal glands even though they're not mating. So nymphs will eat from the glands, other males will eat from the glands, females that have never mated will eat from the glands. So there is a resource for all members of a German cockroach intrusion. And so here's a brief video illustrating this. You can see here that the male raises his wings, and this draws in the female who wants to consume from the Tergal gland. So she will consume the secretions created by the Tergal glands, and while she does so, the male will mate with her. So here you can see their ends were joining and they mated. Early research on the Tergal glands have shown that they contain sugars and nuptial feeding stimulants. So that was found by Nojima et al., that group in 1999. In 2006, Salzman et al. Found or isolated 4 protein bands. And in 2015, Myers et al. Looked at those bands and did peptide sequencing. One of the bands was identified as alpha amylase, which is a substance that is used to digest starches and sugars. And another one was known as BLAGI2, which is the focus of my dissertation. So BLAGI2, it's a 35 to 45 kDa protein. So it formed this 35 to 45 kDa band when analysis of the Tergal glands were done. And so what we know about BLAGI2 is that BLAGI2 is a protein that is found in the wings, the fat body, the egg casings, and other tissues. Before this manuscript, not all of the tissues were really examined. So this paper helps to elucidate the protein content, the BLAGI2 protein content in tissues that haven't been analyzed. And also BLAGI2 mRNA and protein expression profiles in the Tergal glands were unknown before our analysis. And so our paper serves to sort of delve into that. And so the hypothesis, again, is that BLAGI2 mRNA and protein are expressed in multiple tissues and life stages with relatively high expression in the male Tergal glands. The objectives of the manuscript were to determine the relative abundance of BLAGI2 mRNA and to also quantify the expression of BLAGI2 protein in different tissues, sexes, and life stages of the German cockroach. And so here's a pipeline of our first objective. We wanted to look at the gene expression of the German cockroach in different tissues and life stages, and we did this by using qPCR. So basically that is a tool to show you the gene expression, relative gene expression as well of different samples. And so we started out by stabilizing RNA in our samples, then isolating the RNA, then doing reverse transcription and creating cDNA from the RNA, and lastly doing quantitative PCR of our samples. And so our life stages that we used were nymphs, gravid females, which is just another name for females that are pregnant, that their eggs have been fertilized. We also used females, which are females that haven't mated, and males, which also have not mated. And so we looked at the head, the wings, the gut, the 17th tergites. Tergites are just the dorsal abdominal plates. So 17th tergites are the 17th dorsal segments of the abdomen in a cockroach. We also looked at the carcass, the ootheca, the male Tergal glands, the 5th and 6th tergites. And so I will go over these abbreviations later on. And so we looked at these statistics. We did 3 biological replicates and also 3 technical replicates. We used the Liebig-Schmicken method of gene expression analysis. We used a z-score of these values of the negative delta delta C t values that were derived from Liebig-Schmicken. We also followed it up with ANOVA, analysis of variance, and students t-test. And here are our results. So we patterned the results in terms of heat maps. And so the heat maps just show the range of expression. It's actually color coded. So the expression level is based on the z-score of the negative delta delta CT value. So basically, a color of red means that there was high expression of BLA-G2 in the sample, whereas the color blue means that there was relatively low expression of BLA-G2 in the sample. And so here you can see we did it for nymphs, gravid females, females, and males. And for the nymphs, you can see that we included, as I mentioned before, we included adult male Tergal glands as a positive control. You can see that it's the highest expressed BLA-G2, or has the highest amount of BLA-G2 gene expression. But also you can see that the substantive tergites of the nymph also have high expression. This may be due to the fact that we used males in our analysis, in our samples, and it may be that BLA-G2 mRNA localizes to those plates, the substantive plates, in preparation for molting into an adult. And so there may be localization of mRNA there when it comes to preparation for development into adulthood. We also looked at the gravid female. The female whole body had high expression of BLA-G2, as did the male Tergal gland that was included. We looked at the female that hadn't mated, and we found that the whole body and the Tergal glands had highest expression of BLA-G2, and that was the same case with the male. The whole body and Tergal glands had the highest expression of BLA-G2 relatively. And so for the BLA-G2 protein expression, we looked at the Western blot. We did a Western blot of the samples. So here's the pipeline. So this was a follow-up to our qPCR data. We wanted to see how the protein expression of BLA-G2 sort of patterned out in addition to the gene expression. And so we started off with homogenization of tissues. We followed up with gel electrophoresis. We then transferred the proteins from the gel to an extracellulose membrane and incubated that membrane with primary antibody, followed it up with secondary antibody, and then finally detection. And so our outline for the Western blots are as follows. We use the same life stages. We use similar tissues, so head, wings, guts, etc., and similar statistics. So we used 3 biological replicates. We used percent relative intensity. So that's basically how dark our bands ended up being when we visualize them. So the darker bands means that there was more antibody binding to the surface, which means that there was a higher expression of BLA-G2 protein. There's more BLA-G2 protein found. We also did ANOVA and students t-test. And so here we see that our results are very interesting. So let me just go ahead and again illustrate the abbreviations. So N is nymph, GF is gravid female, F is female, M is male, and TG are Tergal glands. And so the results are what we expected for the most part. So what we expected was that we would find a 35 kDa band or a single band that denotes BLA-G2, and that's what we found. But we also found additional bands. We found a 70 kDa band and a 105 kDa band. And so we weren't sure what those meant. And so we followed it up with proteomic analysis. And so here we can just see the bar graphs that show the relative expression. Tergal glands had a very high amount of BLA-G2 expression, as did the males. The gravid females also had a decent amount of BLA-G2 expression as well. Something to also note is that the gravid female has a tertiary band, that 105 kDa band. That is unique to the gravid female, so none of the other life stages seem to have it. So it may have something to do with reproduction. We sort of speculated on it upon seeing that band. It's interesting that the gravid female has it, but the virgin female, the female that hasn't made it, does not express that band in that region. We looked at the different life stages. So the nymph has relatively low expression of BLA-G2. The male Tergal glands that we included has the highest expression compared to any of the nymph tissues. The gravid female has expression of BLAGG2, although the male Tergal gland seems to be higher expressed or expresses BLAGG2 to a higher extent than the gravid female tissues. Note also in the gravid female that 105 kDa band is present in all tissues. So it wasn't some sort of mistake or artifact when we detected it in the whole bodies. We also detect that tertiary band in the tissues, the separate tissues of the gravid female. The female has relatively low expression of BLAGG2. The male Tergal glands included express BLAGG2 to a greater extent than the female individual tissues. And the male has high expression of BLAGG2. So you'll notice that the wings have high expression of BLAGG2 protein. Those were interesting results because in the qPCR analysis of the wings, the male wings did not have a lot of BLAGG2. And so you would expect that if there was a lot of BLAGG2 protein, there may be a lot of BLAGG2 mRNA that ended up being translated into protein. This doesn't seem to be the case. And it may be that the wings of the adult male are just simply absorbing proteins from the environment, from the feces of the male, which contain BLAGG2 as well. And also the wings rest on top of the Tergal glands. So the proteins from the Tergal glands may be absorbed into the wings. And that may be what is responsible for this high protein content that you see here. And so we followed up our results of the Western blot with mass spectrometry of the unexpected gel bands. So typically with an antibody binds to a sequence on a protein called an epitope. That's a sequence that the antibody recognizes and binds to. One band that is detected, if that's the case, then that means the antibody is effective. You don't want an antibody that binds to different things. You want an antibody that is specific for the sequence that you're looking for. But we detected multiple bands. We thought it may be that our antibody wasn't very good and that we had non-specific binding. And so we followed this up with liquid chromatography mass spectrometry that basically allows you to identify the identity and also the relative abundances of proteins in a sample. And so this is our pipeline for mass spectrometry. What we did was we inserted paper tips into Tergal glands. Those tips absorbed proteins in the Tergal glands, and then we homogenized those proteins. We then fed those proteins down a gel and dyed the gel. And so we were able to visualize different bands in the gel, cut out the bands that we didn't expect, and actually dissolve them and use them for mass spectrometry, mass spectrometric analysis. We also did the same thing for that 105 kDa band in the gravid female. So the gravid female also had analysis of its 105 kDa band completed. And so for the pipeline of mass spectrometry, you can see here that the proteins are broken down into peptides. Then high pressure liquid or high performance liquid chromatography is completed, and they're injected into the mass spectrometer, which will detect or quantify the mass charge ratios. The ions are then, or the fragments are then further broken down into B ions and Y ions, which are again inserted or injected into the mass spectrometer, and their mass to charge ratios are found. And then de novo peptide sequencing and protein database search will give you the identities of the proteins as well as their relative abundances, intensities, etc. And so 3 isoforms of BLAGG2 were detected when we did this analysis. So an isoform is just, isoforms are just proteins that come from the same gene or similar genes, but their sequences are a little different from each other. So they may have undergone alternative splicing or some other mechanism that resulted in proteins that are slightly different. They may also execute different tasks, different functions. In a lot of cases in disease, you may find that a different or an alternate isoform is responsible for sickness or contributes to the sickness of the individual. And so here we can see that we've identified 3 different isoforms of BLAGG2. And so all 3 of the isoforms were found in band 1 and band 2, and 2 of the isoforms were found in the 105 kDa band. And so all of this information, the molecular weights and the protein IDs, were derived from GenBank. And so mass spectrometry answered our questions. So it gave us the identities of the bands. So the 70 kDa band turns out to be BLOG2, and also the 105 kDa band turns out to be BLOG2. And so that made things even more interesting. So we developed the idea, or we then concluded that the 70 kDa band is likely a dimer of BLOG2. It's essentially two BLOG2 proteins bound together. Because 70 is a multiple of 35. That was one reason. That was one part of our reasoning. Also, we did a blast of the BLOG2 epitope, the sequence that the antibody binds to, and we found that none of the other proteins detected in these bands had the epitope that BLOG2 has, or anything similar to that. And so we believe that there was actually specific binding of the antibody to BLOG2 going on. We also have previous literature to support our claims. So we have literature from Aruda et al. And Lee et al. Aruda et al. Found that isolated a 70 kDa protein that had its initial sequence, its beginning sequences were similar or nearly identical to BLOG2. And also Lee et al. Isolated BLOG2 dimer. It was complexed with an antibody, and they're able to do that as well. In regards to the 105 kDa protein, we also believe it is BLOG2 due to the fact that it's a multiple of 35. We also did a blast and compared the BLOG2 epitope to the other proteins and found that it was specific. And we also did Western blots of the Uthike, which contains the most abundant proteins that would normally non-specifically bind. And we found that there was very, very low signal, if none at all, in the Western blots of the Uthike, which means that there likely isn't non-specific binding going on, and that it's just simply that BLOG2 is forming a dimer or even a trimer in the case of the 105 kDa band. So my summary and conclusions for objective 1 were that BLOG2, mRNA, and protein are detected and expressed in all life stages, sexes, and tissues analyzed. BLOG2 is highly expressed in Tergal glands and not just a contaminant. BLOG2 forms a dimer in all life stages. Multiple bands are the result of BLOG2 multimers, like dimers and trimers potentially, and not non-specific binding. And the 105 kDa band that is exclusive of the Gravid Female indicates a trimer. It may be a unique function that has to do with the development of the oocytes. So on to chapter 3. Manuscript 2 in preparation is profiling proteomic isoforms in Tergal glands of the German cockroach. So this is where I go further in depth into proteomic analysis. I look at mass spectrometry results of different tissues of the German cockroach, mainly focusing on the adult male Tergal glands. And so this gives you a brief intro into isoforms. Protein isoform, for example, is as I said, it's an isoform that typically comes from the same gene, but has slightly different sequences. And this difference in sequences can allow them to have different structures, functions, or effects on health. Tergal glands are something that we reviewed before. And so BLOG2 isoforms were detected in chapter 2 from the Tergal gland secretions. And lastly, protein-protein interactions and complexes may form as a result of isoforms. So isoforms may come together to form complexes, or they may interact with different proteins and have different modes of action. And so my hypothesis and objectives are as follows. So my hypothesis is that there are multiple protein and allergen isoforms active in the Tergal glands. My objectives are to determine the identity of proteins expressed in the Tergal glands, and also determine the identities of proteins that co-immunoprecipitate or interact with BLOG2. And so this is the mass spectrometry pipeline, again, because I did this research on the Tergal gland tissues and not just the secretions. So what I did for this manuscript were I dissected out the Tergal glands, I homogenized the proteins, broke the proteins down into peptides, did high performance liquid chromatography, injected them into the mass spectrometer in a similar cycle to what I did in the first manuscript. And after that, we were able to identify these proteins. by using the mass spectrometer. So apolipophorine was the most abundant protein. There are also other proteins like actin, 5C, myosin heavy chain, filament A, and BLAGI2. So we found that BLAGI2 was one of the most abundant proteins found in the Tergal glands. Mass spectrometry of the Tergal gland tissues found 3 isoforms, just as mass spectrometry of the Tergal gland secretions found 3 isoforms. And so the isoforms have overlap in terms of the peptides that were identified and sort of tracked to identify them. So for example, the first isoform in pink had 6 peptides. So 6 peptides that were unique to the first isoform were identified. But it also, for the second isoform in green and the yellow isoform, they share peptides. So they have similar sequences in some regions. Some of their peptides overlap, but some of the peptides are also different. So there are 6 peptides unique to the isoform in green and 2 peptides that are unique to the isoform in yellow. And so I did comparison of the Tergal gland secretions and the Tergal gland tissues and found that there are 130 proteins that both the secretions and the tissue share, but there are 173 proteins that were unique to the secretions and 478 tissues that were unique to the tissues. And so in order to sort of parse through this information and find out what it was useful, I did target P analysis. And basically that will tell you signal peptides or what proteins have signal peptides. Signal peptides will tell or sort of give a message that the protein in question needs to be secreted. So basically I wanted to find out which proteins in the secretion evolved to be there, which proteins were transferred to the secretions, because that's, I think, one of the best way to sort of figure out the functions of the secretions and the proteins that lie within are to find out which ones have been programmed or evolved to be there. And so I took all of the sequences in the proteins, or all of the sequences of the proteins identified in the Tergal secretions, and found that only 84 contained a single peptide. So the likelihood probability would just tell you what is the likelihood that a given protein has a single peptide. And so I did a cutoff. Anything over 50%, I claimed had a single peptide, and anything 50% or under, I claimed did not. And so 84 proteins had a likelihood probability of 50% or more. So you can see in the top part of the graph, those are the proteins that contain a single peptide, and the bottom half do not contain a single peptide. I then follow this up with co-immunoprecipitation, and essentially that is a way of finding or potentially finding out the interacting partners of a protein. So ideally what you do is you will couple an antibody to something like magnetic beads and incubate them, and the antibody will bind to the beads, and then you'll introduce your protein mixture, and the protein that is specific to that antibody will bind to the antibody, but also proteins that interact with the protein bound will also couple, so they'll also sort of co-immunoprecipitate. So this is what I did to find out which proteins may potentially interact with BLAGI2. So I used a BLAGI2 antibody, I incubated it. This attracted the BLAGI2 protein, and then other proteins were attracted to the BLAGI2. And after this, you can feed this into or inject this into the mass spectrometer for analysis and identification. And so here's just the pipeline. I did co-IP analysis of the male Tergal glands, the male whole body, so the entire body of the male, and also of the Gravid female. And so I broke all of these proteins down into peptides individually in different samples, and injected them into the mass spectrometer for identification. And I found that there were some similarities. So for example, myosin heavy chain was the most abundant in all of the samples. So the Tergal glands, the male whole body, and the Gravid female whole body had high amounts or relatively high amounts of myosin heavy chain. And this differs a little bit from just the analysis or the mass spectrometry of the Tergal glands by themselves without co-immunoprecipitation. So this means that myosin may in fact be interacting with longitude because it differs from just the most abundant protein found in the Tergal gland tissues. So here you can see some functions. Myosin heavy chain, typically the function is actin filament binding, ATP binding, and motor activity. Apolipophorine was also common, and its function is lipid transport activity and so on. I also did BLAST2GO analysis, and basically this uses algorithms to find out the different functions of all of the proteins in a sample, given that you're able to inject or sort of imply their sequences, their FASTA sequences. And so I did this with the Tergal gland secretion proteins that I found. And yeah, so I was able to deduce the molecular functions of quite a few of the proteins. The most common function was serine type endopeptidase activity. So essentially, a lot of the proteins found in the secretions broke down proteins. Aspartic type endopeptidase was also a very common activity of the proteins. So essentially the Tergal secretions contain a lot of proteins that break down other proteins. And I did this specifically of the 84 secreted proteins. So I did this of the proteins that have evolved or are programmed to actually be inside the Tergal gland secretions, and not ones that just happen to be there due to contamination, or proteins from the environment or the wings, or from cells lysing and leaking housekeeping proteins, or proteins that are on the cuticle from neighboring adjoining areas. So this was more of an in-depth analysis about the functionality of the proteins in the Tergal secretions and why they evolved to be there. So again, just to clarify, so the serine type endopeptidase was the most popular group, and aspartic type endopeptidase was close, the second popular. And here you can also see the biological processes of Tergal gland secretion proteins. So this is a little bit different from the molecular functions because this gives you more of an overarching explanation of their function. And so this sort of goes back to what I was saying before. In this analysis of Tergal gland secretion proteins, proteolysis was the most common function or process, and carbohydrate metabolism was the second most common. So that means that there are quite a few proteins that also broke down sugars and starches, just basically metabolized them as well. So yeah, to reiterate, proteolysis and carbohydrate metabolism were very common. I also did this analysis on the Tergal gland tissues themselves, and I found that ATP binding was the most popular when it comes to the molecular functions. I found that translation was also one of the most popular biological processes in the Tergal gland tissues, which makes sense because I used whole cells and lysed them. And so there are a lot of proteins dedicated to the process of translation because they're coming from entire cells. I also looked at the location of Tergal gland proteins. And so basically it just tells you what part of the cells or what location the proteins are located in mainly. And so the most common region for the proteins was the cytoplasm. And the integral component of membrane was also a very popular region. So that tells you that, right, a lot of proteins were created in the cytoplasm, stay in the cytoplasm, and that sort of tells you where a lot of your proteins might also be shuttled as well. And so my summary of that are that there are 303 secretion proteins, 84 containing a single peptide. And overall, we found that there are 608 proteins in the Tergal gland tissues. Proteolysis is the most common biological process in the secretions. Translation is the most common biological process in the tissues. Tergal glands may function as a source of digestive enzymes, for example, alpha amylase, and 255 proteins co-immunoprecipitate with Blot2 in the Tergal glands and 220 in the male whole body, 207 in the Gravid female whole body. So essentially, what may be happening here is that the Tergal glands have evolved to be a secondary source of digestive enzymes. The most common proteins in the secretions broke down proteins and starches, and because cockroaches live in adverse environments, they live in environments where they really have to diversify their diet. They may have evolved to have a secondary source of digestive enzymes to help them better break down food. This is perhaps why all of the members of a cockroach colony or a cockroach intrusion will feast from the Tergal glands, even when they're not mating. I also found that multiple proteins, for example, NADH, ubiquinone oxidoreductase, and other ligases are found in all samples, but only co-immunoprecipitated in Tergal glands, which may mean that there is a specific process that is going on in the Tergal glands. And so my overall conclusions are that Blot2 mRNA and protein dimer are detected and expressed in all life stages and sexes and tissues analyzed. Blot2 is highly expressed in Tergal glands. The 105 kDa plus band in the Gravid female may indicate a trimer, and multiple proteins interact with Blot2 exclusively in the Tergal glands. So there may be specific reactions going on that are unique to the Tergal glands. Also, the Tergal glands contain 608 proteins, and they may have evolved as a source of digestive enzymes to help break down toxic food or a diverse set of foods in the environment. And so for future research, I'd like to size exclusion chromatography. That will basically tell you if the Blot2 protein forms protein complexes, if different isoforms form different complexes, and that may clue you into the functions of the protein, especially if these complexes are already described in literature. It would give us a better idea of the function of the protein. I'd also like to do RNAi, and that is essentially something where you inhibit the production of the protein, and that will tell us what its function is by the phenotype of the cockroach. And so the RNAi is something I was going to work on, but COVID made it a little bit more difficult. I believe that a possible function for Blot2 in the Gravid female is that it's a pregnancy, it aids in pregnancy, or it has a function in the development of the eggs or the cockroach embryos. Blot2 has similar sequence similarity to pregnancy-associated glycoproteins. They are also inactive aspartic proteases, and they also look quite a bit similar. They have similar structures. And so the pregnancy-associated glycoproteins normally aid in development in the in pregnant ungulates. So for horses or pigs or so on, they typically would use these for fetal development or development of the fetus and the placenta, regulating different activities. And so this may be the case in the German cockroach. It may also use inactive aspartic proteases for development as well. And so for future research, I would like to perhaps use Blot2 and other allergens isolated in the Tergal glands. Allergen-specific immunotherapy is a therapy where you use allergenic extracts or allergens to sort of desensitize people who have a sensitivity to allergens, who are allergic and have an exaggerated response. And so, for example, you can use cockroach proteins to perhaps decrease the ratio of immunoglobulin E to immunoglobulin G4, and that may reduce inflammation of cells in target mucosa, so mucus linings in the nose. And so this would be a very interesting and sort of revolutionary form of research, because quite a few people are sensitized to cockroach allergens, especially in the inner cities. So here's an example of injection with the extracts. Okay, thank you. So I'd like to thank Dr. Maya D. Ganalkar, my advisor, Dr. Michael Scharf, Dr. Uma Ariel, and Dr. Gary Bennett. These are all the chairs of my committee. I'd like to thank them all for helping me and their patience and guidance. I'd like to also thank especially Maya, my advisor, for his patience and his teaching. I'd like to thank the Center for Urban and Industrial Pest Management, the Purdue Diversity Fellowship, the Pi Chi Omega Scholarship Program, Scholarship and Awards Committee, my mentor, Dr. Robert Corrigan, who really taught me about Purdue and sort of gave me more information about what it means to do a PhD and why it would be something that I could potentially do. I'd also like to thank my coworkers and ex-coworkers and friends, Dr. Julius Eason, Dr. Aaron Ashbrook, Cecilia Foley, Jennifer Topolsky, Hunter Clawhorst, and Sarah Stack. And I'd also like to thank my parents for creating me and for supporting me during my PhD. Yeah, without them, this would not be possible. So thank you all. And do you have any questions?